We're not quite full yet, but I'm going to just go ahead and get going so that we have enough time for discussion. So to introduce myself, I'm Nicole Theodosiou. I'm a faculty at Union College in Schenectady, New York. And I'm also chair of the Professional Development and Education Committee at SDB. This is my first year chairing this committee. So it's a great time, a trial by fire here in the day of COVID. So today what we're going to do is myself, and I'm joined by Michael Barresi at Smith College. We're going to sort of give you some tips on how to think about moving your course online. And I hear a bit of background, so if everybody could please mute yourselves. We have decided to record the presentation part of this round table theme table. And then when the presentations are over, we're going to stop the recording so that people can feel free to ask questions. All right, so to get started, I just wanted to talk a little bit about why we're doing this and what our thinking was behind this. So Michael has been hosting these weekly Zoom meetings as a support group, really, for people to share ideas on how to plan their courses for online. And it's been really helpful. So I thought it would be really nice to have a theme table like this. And I was fortunate in that at Union, we're on a trimester system. So on March 30th, I started to teach developmental biology as an upper level course with lab online. And I did the entire course online for the first time. So with that, I learned a few things and going to Michael's sessions. And I've written a manuscript that I'm sharing here on this page about how to redesign your in-person course for online. What we did, most of us in the spring during the pandemic, when we all had to vacate our labs and our classrooms, is take what we normally do in person and directly transfer it to the remote platform. And that's what educators call emergency remote learning. And we didn't really have the time to carefully and thoughtfully design what an online learning environment would be for our students. It was just this panic rush. Now that we find ourselves and institutions making decisions to offer online courses in the fall, we're all under this extreme pressure to do a better job, especially with surveys that are showing on the national level that all institutions should be expecting at least a 20% decrease in enrollments. So with that, we need to start thinking about how can we create successful online courses. And generating an online course is a very, very different skill set than the strategy that you would use to design an in-person course. And a lot of us are really familiar with that in-person learning. And those of us who have taught now online kind of miss that personal interaction you have with your audience when you're speaking to them. So I want to just highlight, I'm going to start by sort of giving some overall approaches to thinking about designing your course for online. And then Michael's going to go into a little bit more detail and also demonstrate what are some key tools that the textbook offers for e-learning. So the just 2 points I want to make today is the first is planning the big picture. A lot of what you need to, or what I'm recommending, is really take the time to think what it is that you want to get across. So I'll talk a little bit about that, and then I'll talk a little bit about how to use group work to generate connectivity between your students, because that's what we really miss in the online setting. In terms of planning for the big picture, I challenge people to think broadly about what it is that you really want your students to get out of the course, realizing that you can't do everything exactly the same as you did inside the physical classroom. So think about what are the big questions in the field. If you're teaching developmental biology, what are the big questions that you want your students to understand? The questions I chose, and I will admit I took this straight out of Michael's chapter 1. He has a list of sort of essential questions in developmental biology. How do cells communicate to form an embryo? What processes control cell and tissue patterns? And how do cells organize into functional structures? I could have chose other questions. I just happened to choose these, as these were areas where I could imagine the course material that I wanted to get across to my students would fit nicely in these big questions. It takes a long time to establish big questions. I teach a senior seminar course on evolution of limbs, and in that course, I actually have only one essential question that I ask my students, and that is, what is the genetic basis for morphological change? And the entire course is constructed around that big question. This is in the pedagogical world called what's it called? I forget now, but it was written up in this book called Understanding by Design by Wiggins and McTighe. They have a website where some really nice tips on how to come up with big questions for your course. But once you have these big questions, then you can start to organize your content. So distill what you normally do and think about the big questions you've established and say, all right, what is the knowledge that my students need to know and understand in order to answer these questions? Okay, so maybe you won't be starting the same way as you normally do. Maybe you will be mixing and matching material in a different way than you normally do. But what this does help you do is distill the material and also maybe get rid of some things that you don't need anymore. When you move to online, I've found anyways, you can't cover the same amount of content. You're going to lose about 15–20% of your normal content material. And by establishing these big questions of understanding, it'll help you decide what is the most important thing that your students need to know and learn. You can also use these questions as a means of assessment. So for example, my final exam this term was only one question, and it was to take each of these big questions and answer them using material from the course. It was completely open book, open note, but it's a really nice way that you're asking your students to synthesize the material, not just regurgitate, you know, cell-cell interactions and signaling pathways. Let's see, in another aspect in terms of designing your assessment, think about, instead of big exams, like a big midterm and a big final, think about things that are scattered across the term, little bites that students can dig into along the way. You might be a fan of weekly quizzes where you're sort of testing recall and basic knowledge, or you may want to do something where you're posing an exam question every other week where students have to apply their understanding. There's also really nice creative projects you can do. And one thing I really recommend is providing a lot of flexibility and choice to the students. You may have your students do a creative piece. Maybe they're going to do a podcast. Maybe they're going to do an infomercial. Maybe they're going to do a video description of something. Or maybe they're going to write a paper. But in order to reach your broadest audience and realizing that some of your students may not have access to a computer, they may be doing their entire course on a phone, maybe they don't have access to internet all the time, giving them the choices and flexibility is really, really helpful. Another big picture thing to think about, once you've established your big questions and you have an idea of what your assessments are going to look like, I also really recommend thinking about what is the workflow and make it very clear to your students. So this is a graphic from my syllabus from this past term. And I wanted to show my students visually what their week is going to look like. So Sunday through Saturday, we're going to have an optional online tutorial with small group discussion on Wednesdays. And then on Fridays, I had a required synchronous class. So this goes to the question of how much are you going to have students do on their own in an asynchronous manner versus how much are you going to require your students to be live in class with you? And realize that students may be in very, very different time zones, and they may not always be able to make required meetings. So here I tried to visually show this. Again, this is really nice to have visuals for your syllabi so students can easily see what they're supposed to do when. And you can see that I have readings, watching videos, and lab assignments as sort of something that they're going to schedule on their own time on the bottom. Okay, so the second point I really want to make is using peer groups to generate connectivity between students. So there was a psychologist, Ignazi, who wrote many books and made the observation that social interaction is essential to cognitive development. So students really need to talk to each other, to work together in order to learn well. And so the use of peer groups, though it can be tricky, is a great way to do this, and you can use peer groups in many ways. Peer groups, and I know Michael will talk more about this, can be done in synchronous classes. So when you have those live classes, you can have students working on case studies together. They can do article discussions. And this can be done easily in breakout rooms and Zoom very nicely. In my courses, I always have students do reflective writing. So they do blog work and they give each other feedback every week. So they're interacting and having conversations online. And for this, I use this as a way of students making connections within the material and between the material we're talking about in class and new topics. So a good example of this would be after a week of discussing just gastrulation and Xenopus, for a blog post, I will post a prompt, and that prompt may be 5 different videos of gastrulating embryos of different species. So zebrafish, chick, sea urchin, whatever I can find. And then I'll ask them to make observations. So, you know, what do you see? How are the cells moving? How are they moving in relationship to what we learned and know about in Xenopus? How are they different? And then why do you think they're different? So in this way, I'm using the blogs as a way of helping them connect and articulate their understandings, but also make connections to new content. And finally, for group work, which is very hard to pull off well, I wanted to talk a little bit about a lab that I designed that involves role playing. And so everybody's a little bit stressed out about how to do labs online. And so I wanted to show you the lab that I did. So this is a little bit of backyard science meets NSF proposal. So I had 24 students and I broke them up into 4 lab groups of 6 students each. And I had the students learning about teratogens. They read Tyrone Hayes's paper. They went to Michael Barresi's website and watched the interview with Tyrone and discussed some of his work. And then I had 4 students who I had pre-screened, who I knew lived in upstate New York near ponds. And I had them go out and find frog eggs. And so this is a clutch of frog eggs that they pulled out from a pond. and they brought them back to their homes and stuck them in salad bowls and Tupperware containers and took care of them. And so the graduate students, so every student in the group was given a different role, a graduate student, postdoc. I had chief equity officer to make sure everybody was working well together. I had a chief understanding officer to make sure everybody was understanding what we were doing and why. A grant organizer and a grant editor. So in this group, everybody was working on a project to identify a teratogen in the graduate student's home. And then they had to do a literature search on that teratogen, and they had to design an experiment. The graduate student had to execute the experiment, collect the data, and then hand it off to the postdoc for analysis. And so in this way, every student had a very distinct role to play in this big project. So the students did the experiments, they analyzed the data, and then they used the data from the backyard science experiment to write an NSF proposal. And part of that proposal process was multiple steps. They had their preliminary results, they did a proposal outline, and they exchanged it with other groups to get peer feedback. They then wrote and submitted a proposal, and we ran study sections. So I gave them an NSF scoring matrix. And part of this was also learning, most essentially, how to work well together. And then also how to design of experiments with the tools you have. If you're in your home and all you have is ibuprofen or a bottle of vodka, whatever it happened to be, then that's your teratogen, right? And then how do you decide how to make a dilution? Well, I have a 2-liter empty Coke bottle, so I'm going to use that, you know, to do my dilutions in. So the students had to be really creative and find their own solutions. And in many ways, it sort of mimics what we do in the lab, in that we don't always have all the available reagents and resources we want for our experiments. And then we sort of modify and troubleshoot as we go along. So that's what I wanted to sort of share with you today, the idea of creating big questions to answer and building your content and assessments based on those big understandings, and then using group work to bring students together and getting them working well together. And before I hand it over to Michael, I just want to make a quick plug that as the chair of PDEC, we do have an opening available. And if you're at all interested, please reach out to me, send me an email, and I'd love to have any of you on my team. So thank you, and I will stop my screen share. There you go, and hand it off to you, Michael.
Well, thank you very much. That was great, Nicole. I always learn something new every time I hear what you're up to. I also want to applaud the amount of work you did in such a short period of time in the midst of what was happening in the spring to actually collect data and assess how your students were learning. Like, if there's not a better teacher, I mean, I don't know, and that was fantastic. So let me share my screen. 1 second. Okay. Can you all see one screen? My one open PowerPoint? Okay.
Yes, I see it.
Thank you. So I just want to start off with, I know that this situation that we're in can bring us down a little bit and get us stressed out. But I really do believe with everything I have that what it's forcing us to do as educators is going to make us fantastically better teachers in the long run. I really do. So I really hope you all embrace what is going to be happening next year and push your teaching perhaps a little bit more in creative ways than you wouldn't have had to before. At least at my own institution, I have a lot more, or all the faculty have been given a little bit more leeway, a little bit more flexibility to try new things. I think this is a great time to do it. And I'll also say that, you know, for years, we've been trying to get faculty to think about what they're doing in their classroom to really foster learning and push the roles of active learning while they're doing it. That's a hard thing to do. But this transition is now forcing a lot of us to put what we would perhaps consider doing in the classroom in another space, in a virtual space for which students interact with autonomously. And now it's forcing us to reimagine, what are we doing when we are together in that virtual or physical classroom? It has to become more active. So it's going to be a lot of fun. And I'm really only going to try to talk for as little amount of time as I can, maybe 10 minutes. I'm going to go a little fast, but I want to draw your attention to a webinar that I produced in the spring that goes into a lot more detail on everything I'm going to talk about. And the link to that is right here. And again, in the chat window, if you showed up a little late, I posted a file of this whole PDF. So any links in here will be directly in that PDF if you download that file. I'd also like to start off with what some of my overall guiding recommendations are that frame how I build a given course. And particularly in this time, as Nicole mentioned, less is definitely more in this case, and really thinking about the big picture of what concepts are key. And those opening questions were really good. But how else can we be guided as instructors to thin down our course? There's so much information, and it's really hard for us, and imagine for our students, to distill it to what's really important. And we've done some things in the latest 12th edition that I hope helps faculty to actually see and make it easy to reduce content, and I'll discuss that. Building that well-organized syllabus. I love the picture idea. Put that right in your syllabus of what a typical week would look like, and stay and maintain that structure. I'll also note that as you're beginning to think about developing your syllabi, with this upcoming fall, it could be unpredictable, right? Some of us are now forced with what's called a multimodal approach, which I would argue we've always been promoting a multimodal, meaning different ways of teaching, which would help a diversity of students and how they learn. But in this context, it's like maybe some in-person mixed with some remote. Now, if we are forced to completely going remote at some point in the semester, we will have been thankful if we had some period of time that's in person. So my point for mentioning it right here is that you strongly consider emphasizing what you can make the most of that in-person interaction, and really foster and cultivate good relationships with those students, which then, if we do have to pivot completely remotely, would hopefully help to carry you through that period of time. But keeping it structured will help everyone. Provide assessments that have integrity, the sort of cheat-proof assessments, and I'll talk about some ways to do that. Provide assessments that empower students in what they're doing and really foster that motivation. What I mean by that is, you know, for all of us who taught in the spring, it was really hard to keep student motivations up. I'm sure you would agree with me. And so think a little bit harder about your assessments and ask, are there any ways that I can make this assessment be student driven? Allow their interest to really foster the direction in which that given project, et cetera, goes. The more you can do that, it'll build greater motivation for those students and result in a better learning outcome. Make a majority of those learning opportunities asynchronous. So again, different time zones, variable internet access. So we're going to have to think creatively about our asynchronous. So those things that you're doing, if you don't know what we have a lot of terms: multimodal, synchronous, asynchronous. So the asynchronous, just so you know, is like content that is being reviewed and studied by students in the absence of you. And in the spring, it was very abrupt. We all went to it, and you know, we all did our little Zoom recordings of us giving our standard PowerPoint lecture. Moving in the fall, I'm going to challenge you all to think a little bit more creatively and say that perhaps that's not entirely good enough. That how do you maintain student engagement during those asynchronous times? You know, I would even admit that probably an hour-long recording of the most brilliant lecture I can give may not be the most captivating thing, right? So really think carefully about what that content's going to look like. Yeah, and students may have to complete a lot of it on their own. And then lastly, what are you going to be doing in that classroom space, that sort of synchronous live opportunities that you have with them? What are you doing to make it active and engaging? So we can divide it up in sort of these three little bins: our sort of asynchronous learning content, our optional live tutoring, and what are those live sessions going to look like? There's going to be a lot of good, robust tools that you have available to you in the ebook associated with developmental biology. We have recorded dev tutorials that should help support some of the lectures that you would be making yourself. Helping the sort of live tutoring sessions are going to be ones that are going to be very advantageous, particularly for students in different time zones. You may be delivering a synchronous activity, but it's not going to work for everyone. You're going to want to consider these sort of office hours at different times to be able to connect with students one-on-one, hopefully, or at least in small groups, again, to foster that relationship and keep tabs on how they're doing. For the synchronous activities, this again, I think we're going to have a lot of fun here in this space now. You're challenged, you're being forced to really reimagine what you're doing with them in that space. And I would push and encourage you to consider the case study problems that we've created associated with the textbook that are designed for an in-class activity. They're interrupted cases, break through, and then you take advantage of the Zoom breakout rooms. And I'll talk a little bit more about that, along with other polling software like Poll Everywhere that can help you keep tabs of student understanding as that session's going on. Some autonomous assessments and overall assessments to be thinking about. So probably many of you are going to be engaging students in that literature, right? As Nicole mentioned, the grant proposal is a fantastic example of a very long assignment. Mine, in fact, lasts a whole semester. And you break it up into the individual drafts. You have a culminating event where you have a study section at the end. It's really a fantastic experience. And I think in this environment, I would even push you to consider collaboration. And I mean that in its literal sense. What if courses, you and other faculty from around the world collaborated together? What if all of us, or whatever, some of us were doing a grant proposal? And in fact, instead of my students reviewing each other's grants, my students are reviewing your students' grants. That kind of interaction is, again, a little silver lining that we may be able to take advantage of in this time and have a lot of fun doing it and learn a lot. Self-scheduled exams, and many of you are going to do quizzes or exams. I definitely second Nicole's suggestion of perhaps smaller group assessments, smaller level ones. But how do you make those sort of cheat-proof in this online world that students are living in? And my recommendation is going to be to do these sort of fictional problems. Again, a kind of case study model, but creating these scenarios that is pushing development the next step into an unknown area that students have to apply what they've learned. There's really no way to cheat on those things. And I'm in the textbook provided all of my initial case study scenarios for the exams. So again, my couple key recommendations is reduce your content. And one thing about the 12th edition that I really actually tried to do this for is to make it easy for you as a professor to literally put in your syllabus. You're only required, a student is only required for a portion of that material. And so I've partitioned it into an essential content, and then these other areas that are called further development. There's further development in the textbook, and there's further development online. These sort of represent levels of complexity. And you can pick and choose what you want them to show very easily, and because it's all compartmentalized in this way, it actually makes your life a lot easier for easily reducing content where necessary. So I hope you take advantage of that structure. Moreover, a lot of us are going to be pushing students to move into the research literature, particularly in this environment. interacting with that, looking at and reading and analyzing data in the primary literature is critical. In the 12th edition, especially the ebook and online links, we have a very robust now direct guide to help students learn how to find research, how to get through that, get into the literature, go directly into PubMed, how to break that search down. And when you get an article, how to break that article down. What is it? What are the parts of it? What do they mean? So we literally have a guide to help your students, and I'm really proud of it actually, and I hope you take advantage of it. You assign a student a paper to read. And as Nicole mentioned with the Teratogen approach, her students are reading papers. And then they went to my website and looked at a web conference with Tyrone Hayes, and that supplements that and brings it to the next level of students working with it. We have over 60 of these recorded, freely available on my website. Moreover, in terms of the assessments, I am sharing my sort of fictitious case studies that I provide in exams. So these are scenarios broken down that ultimately for students to develop hypotheses as to how these things could possibly happen, they have to apply the concepts of development. And again, it's just nothing that a student can cheat on. I have them completely open book, completely open world. It doesn't matter because they're not going to find answers anywhere. For your asynchronous content, again, I hope you will have used and will continue to leverage the dev tutorials, those video recordings that Scott and I have done. We're proud of these. They have animations into them. It's not just a talking head. And students have, at least in my experience, really found them quite foundational to then when they enter into the classroom, have some knowledge that you can rely on to then start asking them to apply it. And they would apply it in the synchronous live sessions with perhaps the case studies that we also provide through this textbook. So in the e-resources, we have PowerPoint interrupted case studies for a variety of the chapters for which you can take, change, modify to make it work for you. But it's a great way to have a problem-based discussion in your class. And then utilize Zoom breakout sessions to have them go into small groups. And what I'll also add that I found extremely helpful is I would also have a shared Google Doc going simultaneously. In that Google Doc, I would have sections broken in, so tables for every small group that was being constructed. Those small groups, when they're working in them, would have to fill out questions in those tables associated with the case. It was a great way for me to monitor all the groups simultaneously and keep track, and then I could jump into individual groups where they needed help. So there's a lot more to talk about. Again, go to that webinar that I've put, which is much longer. I will also mention that SDB has a shared Google Drive that a lot of resources are being put into, and that's that link here. We have a shared Google Doc built into there, again, where people have been putting a variety of resources. As mentioned in the spring, I had weekly Zoom forums with amazing people coming and sharing their challenges and solutions to what's going on in the world of teaching developmental biology. Right now in the summer, we have one coming on the 20th focused on labs, and then one on the 17th focused on relationships and fostering that teacher-student relationship. These are the links to those sessions. I hope you'll join. During those spring sessions, I asked faculty to answer a quick little survey. Again, I provided the PDF of this. I'm not going to go through all the data, but it just gives you some indication of who were the variety of people and places in terms of the students that are participating, the faculty, and the size of the classes was really diverse. And then I just have some specific questions about what was the preferred tech used. So these are some word clouds representing some of the breakdowns of those survey results. You can go through them, see what little words come up. For me, I have experience with, of course, Zoom, these dev tutorials, and Perusall for PDF annotation, collaborative annotation. There were some that didn't work for a lot of people, and they're mentioned here. What about for synchronous instruction? I have experience with, again, these sort of breakout rooms, tutoring sessions, poll everywhere. but go through these, and these could at least give you some indication of what other people were using. For asynchronous content, you have your Zoom recording lectures and your dev tutorials, but I'm going to ask you to push these farther. This is what people did in the spring, and now we're going to need to adapt this and actually raise the standards a little bit of what we're going to be expecting for ourselves and our students. For assessments, you know, a lot of people were doing a variety of things. It's a really great, huge sort of mess of different things here. Quizzes, exams, but case study exams is where I have the most experience, so I'm happy to answer any questions about that. And then lastly, for sort of science writing sort of stuff, a major one was grant proposals. And I do hope that some of us take me up on the challenge of trying to be more collaborative with what we're doing. So with that, I will stop there and really hope that you come up with some questions for Nicole and I to address more specifically that you're concerned about.
Yeah, thank you, Michael. And I have to say, I learned about the resources with the textbook from Michael, and I use a lot of those DevBio tutorials, and the students love them. And it saved me from also having to do similar recordings for myself. Don't be afraid of curating what's available on YouTube and Venmo and everything else. And there's so much available. I had 3 lectures from Richard Harland on gastrulation and setting up the body access. It was fantastic. So you don't have to make everything up yourself as well. You can borrow, steal nicely, and share, because people do really want to share and help out. So there's quite a few of us. We're going to take questions or talk about anything you want us to talk about. If you'd like, you can raise your hands and we'll call on you.